Welcome children. I will be taking in this video enzymes, nomenclature and classification. First, let us deal with the nomenclature or naming of an enzyme. There are three parts for an enzyme: the prefix, the name, middle name, and the suffix. The prefix indicates the substrate, the middle name indicates the reaction it catalyzes, and at the suffix ASC. From the very ASC ending, you can identify whether the biomolecule is an enzyme or not. See, we have the prefix thus indicating the substrate, the center name indicating the reaction catalyzed, and suffix having ASC ending. So, some examples: glucose isomerase. Here it indicates that glucose is the substrate, isomerization is the reaction, and ASC is the ending suffix which indicates it is an enzyme. Another example: pyruvate decarboxylase. Here pyruvate stands for the substrate, the middle name decarboxyl stands for the type of reaction, that is, it is catalyzing decarboxylation reaction, and the ending ASC. So, most of the enzymes have followed this convention with the prefix and suffix, and the name indicated, the main name which is at the center, indicative of the reaction it catalyzes. So, you have a clue of the substrate, you have a clue of which type of reaction it is catalyzing. But there are still some enzymes going with the older convention, where you never get any clue of the substrate it is catalyzing or the reaction being catalyzed. For example, the digestive enzymes like trypsin, pepsin, renin, we have no clue of the substrate or no clue of the type of enzyme, enzymatic reactions it is catalyzing. At the same time, it is not having the ending ASC, instead it is having the ending sin or in ending. Only it is the case of some digestive enzymes. So, that is all about the nomenclature. It is very simple with three parts: prefix, middle name, and suffix. Now, let us move on to the classification of enzymes. Enzymes are broadly classified into six classes. The first class are oxidoreductases, second transferases, third hydrolases, fourth lyases, fifth isomerases, and the sixth class ligases. So, now we will see the reactions, or basically what does each class indicate, which reaction, and I am going to explain with symbolic representative, that is a type of mnemonic. Oxidoreductase. So, the role of oxidoreductase, they catalyze oxidoreduction reaction between two substrates, and it is represented mnemonically or symbolically by this: A oxidized plus B reduced are the two substrates, which becomes A reduced plus B oxidized. We are learning in chemistry. So, all what you need to remember is the reaction and the symbolic representation. So, oxidoreductase catalyzes oxidoreduction reaction between two substrates, and it is symbolically represented by A oxidized plus B reduced, produces two products where A becomes reduced, A reduced plus B oxidized. Second category, transferases. Transferases catalyzes the transfer of a group between two substrates. So, again we are continuing with the mnemonic A and B. These are the two substrates A and B, and there is a group attached to A. Let it be X group. The role of transferase is it transfers the X group from A to B. So, we get the product A plus B bonded with the group. Clear? The symbolic representation or the mnemonic is clear. So, once again, Transferases catalyzes the transfer of a group between a pair of substrate A attached with a group plus B. And what happens in the reaction due to transferase? The group is transferred from A to B. This is the product. Next, isomerase. You are well aware of isomers. They have the same chemical formula but different structure. It catalyzes the conversion of a molecule of isomer into another molecule of isomer. For example, A bonded with B is an isomer. It gets converted to its isomeric form, B bonded with A. So, the atoms present in each elements is the same, but the rearrangement, it has rearranged, this molecular rearrangement taking place, and we get its isomer. A very good example is glucose 6-phosphate becomes fructose 6-phosphate, and the enzyme, using the nomenclature, we can name the enzyme, which is a substrate glucose. So, glucose, and which is the reaction? Isomerization. So, glucose isomerase. So, we have once again, this is one isomer and this is the other isomer. One isomeric form is getting converted into the other isomeric form with the help of isomerase. If this is glucose 6-phosphate, it gets converted into fructose 6-phosphate by the enzyme glucose isomerase by the structural rearrangement of atoms within the molecule. Next, ligase. It catalyzes the linkage together of two compounds. For example, A plus B gives AB. Very simple. So, ligase combines the two substrate A and B, and we get the product AB. A very good example is the DNA ligase, which we will be learning in 12th standard. I will just mention it here. DNA ligase. Fifth category, hydrolases. It catalyzes hydrolysis, that is cleavage of chemical bonds by the addition of water. Again, I am going with the same substrate A, B. There is a bond between AB. On addition of water, which is going to be written as HOH, what happens is the bond between A and B is cleaved, broken, and we get two products, AH plus BOH. See the symbolic representation? Once again, A is bonded with B. It combines on addition of water, HOH. On addition of water, this bond is cleaved, cut. And this B combines with OH to form one product called BOH, and the remaining A and H combines. So, two products are obtained. So, here by hydrolysis, addition of water, the bonds are cleaved. Next, sixth, lyases. It catalyzes the removal of group from the substrate, leaving double bonds between the substrate. Here I will go with two carbon compounds, which are single bond by a single bond, single hydrogen bond, and they are joined to two groups, functional groups. Let this be X and the functional group attached to this C be Y. Now, what happens here? It is a removal of the groups. So, the two groups are going to be removed, X and Y are going to be removed. X and Y are removed and they form one product. They get combined with the bond and they form one product, plus the second product will be the two C's. Now, when this bond is broken and when this bond is broken, what happens is a double bond remains It's left a double bond between the two C's. Clear? So, once again to explain lyases, it's well explained with the carbon compounds. See, we have two carbon atoms attached by a single bond and the other with a functional group X and Y. What happens is the two functional groups are removed. So, the main function of this lyase is to remove the groups. The two groups are removed. So, the two groups are removed and they form one product, and the carbons together form the other product with a double bond. So, this is the action of lyases. So, once again we have 6 categories of enzymes: oxidoreductases, transferases, isomerases, ligases, hydrolases and lyases. You should know its function and the symbolic or the mnemonics against each. And only in 2 places I have cited example in the case of isomerase, because we have learnt it in plant respiration. Glucose 6-phosphate is converted into fructose 6-phosphate with the help of the enzyme glucose isomerase. And in other place also I have explained or given an example with respect to ligase, which we will be learning in 12th standard, DNA ligase. And the most simplest of all these combination is ligase. It just glues 2 substances together like the glue. A plus B gives AB. I hope you have understood today's concept on enzymes, the naming of enzymes as well as the classification of enzymes. Thank you.